right, so we are going to cover part of 10.3 in this video. We've got gene pools and speciation, but this one specifically, we're going to be looking at gene pools and allele frequencies. So these first four questions to consider. So we're going to be looking at the definition of a gene pool, what is an allele frequency, and how they can be used to determine if a population is evolving or not, and what the conditions would need to happen for a population to not be evolving. And then finally, there's a couple other mechanisms of evolution that we'll look at called genetic drift and gene flow, and how those can contribute to changing allele frequencies. Okay, so let's go to this presentation, and just wanted to let you know the modern synthesis is the fact that, since Darwin's time, we have come up with lots and lots and lots of evidence for evolution. And so it's not just the evidence that he put forth, it is a whole body of evidence. And part of that has to do with population genetics, and especially now since we can actually look at the DNA of different organisms. So population genetics, what we're focusing on right now is how populations change genetically over time. And we can measure those changes using, looking at the amino acid sequences, at the nucleotide sequences within a gene, or at the larger genome level. Okay, but the modern synthesis is looking at populations as units of evolution. Okay, so first of all, since we're looking at how a population changes over time for evolution, we know that it's a group of individuals of the same species living in the same place at the same time, right? And so populations are localized groups of individuals, and so they can interbreed and produce fertile offspring. The gene pool of a population is all of the genes and all of the alleles of all of the genes in a population at any one time. So if we see a change in the gene pool significantly after many generations, then we could actually measure whether evolution is happening or not. So it's all the gene loci and all the individuals. So all the genes, that includes all their alleles. So in any population, if gametes are contributing to the next generation, right, with sexual reproduction, we shouldn't see the change of allele frequencies. So if this was a population of flowers, I think we'll see this example quite a bit, where you have a red allele and a white allele. If you had 0.8 of the red allele and 0.2 frequency of the white allele after, alleles get resorted, the gametes produced should be the same, right? Statistically speaking, they should be the same proportion. So we did that with our last, activity with the class. We see after generations, you should see the same frequency of alleles one generation to the next. And this is just showing you an example. If we have one generation where half of so we've got these two pure breeding flowers, right? Here's their offspring. So all the offspring in this generation too are pink. And so we know that 50% or 0.5 of the red and 0.5 of the red. So even when these mix out and sexual reproduction happens, you might get a different genotypic frequency or phenotypic frequency, but again, you have the same frequency of alleles generation after generation after generation. So you should see this. If this population is not evolving, the frequency of these alleles should stay the same. So we have 50% and 50%, whatever the mix may be. This is showing that it doesn't have to always be 50-50. If the population has a higher frequency of red alleles just by chance than it does white alleles, then you'll see generation after generation, you'll still end up with the same, 0.8 and 0.2, like I said, after many generations. We did that in our second scenario when we were looking at the pocket mouse population, where we started with a higher number of one of the alleles than another, but still in the next generation, you would still have that higher number of alleles because there's a higher probability of those becoming the egg and sperm that get fertilized. There is an important theorem that is used in population genetics, and that's the Hardy-Weinberg Theorem. And that's essentially putting a name on what I've been talking about. The Hardy-Weinberg Theorem describes populations that are not evolving. So again, it's stating that the alleles and genotypes in a population's gene pool will remain constant from one generation to the next, as long as it's just Mendelian segregation and so on. And so if a population is in what we call Hardy-Weinberg equilibrium, it means that those allele frequencies are staying constant from generation after generation. There are a few conditions for that to happen, though, because for those allele frequencies to stay in equilibrium where they're not changing, you have to have a large population. We're going to look at this a little bit more when we look at genetic drift. But if you remember, your individual cards, when we were doing the allele changes, your individual population might have shown a little bit of a frequency change, like in that first simulation. But when we looked at the total population of all the class, with all of those simulations happening at once, it was much closer to that 50-50 because we started with 25 of the white and 25 of the black cards, right? And so after you did your simulation with just, 25 and 25, you might have gotten, maybe 47 and 53. But when we looked at all the class data together, so a much larger population, it was much closer to 50-50 for that second generation. And that's the same theory as if you flip a coin, 5 or 10 times. If 7 or 8 times it comes out heads, it's not too crazy. But if you were to flip it 1,000 times, then it's more likely going to, be around that 500 and 500. Okay, so a large population is more likely to be in Hardy-Weinberg equilibrium. You can't have gene flow. You can't have new genes, organisms with new genes coming to the population, and you can't have a significant number of your population leaving either with a certain, allele that's more common to leave or not. So you can't have immigration and emigration happening. There can be no mutations, because if there's a mutation, then you've got a new allele. Mating has to be random. So, males and females can't pair up based on some sort of allele choice, some sort of phenotypic choice. So, like the white mice couldn't only, be looking for white mice. So we can't see some sort of sexual selection, like the females prefer the white mice to the darker mice or something like that, because then we would start to see, selection happening. And then lastly, there can be no natural selection. So there can't be a selective pressure on the population, because we saw what that looks like in that last scenario. When you have a selective pressure, then that will start to change the allele frequencies. So if all 5 of these conditions are met, it's possible for a population to be in equilibrium, meaning the allele frequencies will not change generation after generation. But if these conditions are not met, there's likely going to be some sort of shift in the allele frequency. We're going to explore a couple of those in a bit more detail in a little while. Okay, so this is just showing you a population where you do see the allele frequencies change. So here we have 0.7 and 0.3 to start. And it looks like in this generation, the individuals that survived more frequently than others were these pink guys and the red. And so we kind of had a selection against some of these red ones. And then, but in this next population, it looks like the red ones were the ones that were selected for to mate. These are the only ones that mated. And then eventually, you see that in the third generation, you have only red. So this is showing evolution after it should happen after many, many generations. It usually won't happen after one, but, dinosaurs go extinct. So anyway, it probably isn't going to happen after just one generation, but this is just kind of showing you how you can have those allele frequencies change as certain individuals leave the population. Okay, so how do we get this? If we know natural selection can cause that. So if for some reason there was something here that was an advantage to the red flowers, well, then you can see that happening. So with natural selection. But there's other mechanisms of evolution that are important. And those are called genetic drift and gene flow. Genetic drift has to do with those little populations, like you had your own little population when we were doing the mouse simulation activity. And so, just by chance, just like flipping a coin, some individuals might end up surviving that have a higher frequency of one of the alleles that we're looking at in particular. And so that can start to reduce genetic variation. You can start to see a change in the allele frequencies just due to random chance. And of course, the smaller the sample, the greater chance that has of affecting the overall allele frequencies. There's a little animation that you're going to watch after this on these green and brown beetles living on an island that are susceptible to genetic drift. And genetic drift can happen in some ways. One, it could just be the fact that the population gets really small. It gets forced into a bottleneck. And what that does is it really changes the gene pool. It makes it a much smaller gene pool, fewer alleles. This is just an example here showing of these prairie chickens in Illinois in the United States. The original population has a really vast, you can see there's 3 different alleles probably for whatever gene it may be, has a vast gene pool. But then when that population gets killed off, you can see the range, the habitat destruction resulted in just a couple of small little patches of their population. Well, out of all the ones that died within that time period, you can see the ones that ended up surviving, and it could just be by chance because look, they mowed down the forest, right? That there was only 2 alleles left. And in fact, this blue allele, whatever it represents, is much more common. So the bottleneck effect can result in a smaller gene pool. Generally, there's going to be just one allele that's, one allele that ended up by chance surviving, but it didn't have to do with selective pressures. And of course, the smaller the gene pool, the more susceptible that they are to extinction, because if there's not as many genes to select from, if there's a new selective pressure, the chances of them surviving that as a population might be less. Another way we see genetic drift happening is through the founder effect, and that's when a population might become isolated. And we're going to look at that a lot more in speciation in the next couple of videos that we have from this unit. But if the population ends up being isolated from other populations, like a small representative, and that small representative population has a different allele frequency than the original population, then as that population is under different selective pressures or whatever that may be, you'll see that might just by chance have a higher frequency. We see that with a lot of diseases and disorders that might have, popped up in one region, and if just a couple individuals end up migrating to a new area that have a higher frequency of that genetic disorder, then you can see in a certain area that genetic disorder might be a lot more frequent than it would be in the normal greater population. And gene flow, the last other mechanism of evolution, so genetic drift, basically shrinking down if the gene pool changes due to random events, and probably going to happen to a smaller population more frequently than a large one. But you can also get immigration, emigration, this gene flow happening. So that's when you get fertile individuals migrating to or from a population. If in a larger number, there might be one allele or a different allele that either leaves or gets introduced into the population, that can change the overall frequency of alleles. We see that in human populations. We were isolated in all different corners of the globe. And now with a lot more ease of travel and migration, we've seen a lot of gene flow changing the makeups of populations. Okay, so I am going to just quickly outline this. The mechanisms for evolution are natural selection, genetic drift, and gene flow. And these are 3 ways that we can see the allele frequency changing over time in a population. And natural selection, we know if there's a mutation, that's random. But of course, after that, it's not random. It is absolutely selective pressure will select one allele over another. Where genetic drift and gene flow can somewhat be random, especially genetic drift for sure. And so if you have that small population, you can see alleles change a little bit more frequently. You'll see a little animation on these two, or actually on all these mechanisms right after this.